Hello students, so welcome back to our biology class. We are going to start our class. I'm Dr. Divya from TeenEinstein. So now we are in the second chapter, right? Sexual reproduction in flowering plants. In the last class, what and all we have learned? As usual, just a summary of last class I'll give, so that it will be easy for you people to get an idea. So we'll start the sexual reproduction of flowering plants. So flowering plants, that means angiosperms, right? In angiosperms, flowers are the sites of sexual reproduction. In the last class, we explained everything clearly. So just I'm giving a summary of last class. The angiosperms, they are the sites of sexual reproduction. So in sexual reproduction, what is happening? Male and female gametes, they are fusing together to form a zygote, isn't it? And the sexual reproduction, that includes several steps. So we have categorized all these steps as three major events, right? Three major events are there. Which are these three major events? Pre-fertilization, fertilization, and post-fertilization, right? So the sexual reproduction includes so many steps, and all these steps we have categorized as three major events: pre-fertilization, fertilization, and post-fertilization. Okay. Then we know that sexual reproduction is a complex process and it is taking so much of time. But the duration of this process, it varies in each species, right? Organism to organism, there is variation for the duration of the sexual reproduction process. So compared to asexual reproduction, this sexual reproduction is a time-consuming and complex process. Several steps are included. So it is a complex process and it is taking a lot of time to complete. And we said in angiosperms, flowers are the sites of sexual reproduction. What does it mean? That means all these pre-fertilization, fertilization, and the post-fertilization events, all these are happening in that flower. So flower is the site of reproduction in angiosperms. Then we were discussing about the flower, structure of a flower in the last class. So flower is having four major parts. Who's that? Shiva Karthik. What about others? They can hear? Other students, please respond. Can you hear? Okay. So flower is having four major parts. Which are the major parts? Calyx, corolla, androecium, and gynoecium. These are the four main parts of a flower. Okay. That calyx means sepals. If you are having an hibiscus flower, if you are that hibiscus flower, green part where you are holding that fused part, that is called sepals, calyx. That is fused in hibiscus flower, right? All are together. Sepals are not separate. Then corolla. Corolla means petals. Each petals together, it is called as corolla. Then androecium. Androecium is the male reproductive organ, right? Or collection of stamens. All stamens together, it is called as androecium, the male reproductive organ. In that, stamen is the reproductive unit, a single unit, that is stamen. Together, all, see, one flower will be having 3 stamens, another one will be having 6 or 7 stamens. Number of stamens will be different in each flower. So these stamens together is called as androecium, and the gynoecium is the female reproductive organ, right? So in this, calyx and corolla, they are the non-essential whorls. Which are the non-essential whorls? Calyx and corolla. They are called as the non-essential whorls because without calyx and corolla also sexual reproduction is possible, right? For sexual reproduction, what and all you need? A male and female part. Now the other whorls, androecium and gynoecium, they are the essential whorls of a flower because they are the male and female part. Without that, sexual reproduction is impossible. So essential whorls are androecium and gynoecium in a flower. This sepals and petals, that means calyx and corolla, they are the non-essential whorls of a flower. Okay, clear? Then we were
Yes,
discussing
ma'am.
about the structure of stamen. Okay, in stamen, a single stamen consists of the down, the filamentous part, and the terminal position. At the terminal position of the filament, anther will be present. Okay, down that filament will be there, and on top of that filament, anther will be present. And coming to the anther, anther is a bilobed structure that will be having two lobes, right? So each lobe is having two theca. What is that? Each lobe here. This is the structure of anther. Anther is having two theca. This what is that? This covering like or this section that is called as theca. Okay. So each lobe is having two theca. This is one and this is one. And when we are taking a TS of an anther, it is having four corners, right? Four corners or four sides. Here you can see 1, 2, 3, 4. Four corners it is having. So each corner is having a microsporangia. Here, this corner is having one microsporangia. Here down also, and like that, both lobes will be having microsporangia. So altogether, four corners are there. In each corner, it is having a microsporangia.
Ma'am,
Okay.
4 microsporangia.
Sorry.
Ma'am, 4 or 2? Microsporangia இருக்குதுங்க ma'am. 4 or 2?
Two lobes. See, this is one lobe. I'll show you the structure clearly. See here. This is the structure. That is the time of dehiscence. Okay. Here you can see the structure. Microsporangia means that sporangia, the sac-like structure which is bearing spores. Okay. Don't get
Okay.
confused. That structure, inside the structure you can see, in four corners, that is microsporangia means microspore bearing structure. Inside
Okay.
that, microspores will be present. So in each corner you can see. Okay.
Okay, ma'am.
Here, this is the section. This picture which I showed, this is the time of dehiscence. The pollen grains are coming out. Okay. So it will get fused that time. These separations and all will go off. This is the exact structure. When in board exam they are asking you to write the structure, you should write this one and explain. You should draw this structure and explain. Got it?
Yes, ma'am.
Okay. So each lobe, two microsporangia will be there. In one lobe, two microsporangia. Then the structure of microsporangia we learned. A microsporangia, it is almost round, not a complete round structure. It is almost round. Okay. Here, the structure I'm saying. See, it's not a complete round structure, almost in round shape. And the outer layer is called as epidermis. Okay, this and all we explained in last class. Epidermis is the outer layer, and next layer is endothelium. Endothelium is the next layer. Next to epidermis, endothelium will be there. And next is middle layers. We are not saying middle layers, because it is having multiple layer of cells. 1 to 3 layer of cells will be present in middle layers. And all these layers, they are having protective function. They are giving protection to the pollen grains. Okay. And they are helping in the dehiscence of anther. For dehiscing this anther, these structures are helping. And the innermost, after all this, epidermis, endothelium, and middle layer, next is tapetum. Tapetum is nutritive, it is having the nutritive function, or it is providing nutrients for the growth of the developing pollen grains. So tapetum is having or it is doing the nutritive function, nutrition. That is the function of tapetum for the developing pollen grains. Okay, it is giving nourishment to the developing pollen grains. And in a young anther, at the center of microsporangium. See, again, don't get confused. At the center of microsporangium, this is the microsporangium. And at the center of microsporangium, a group of cells, group of compactly arranged cells are present. See, these are the cells which are present at the center. Okay. And these cells are homogeneous. Here, inside this and all tapetum, this layer. Okay. Inside that, here the marking is there, microspore mother cell. Can you see that? Microspore mother cell. They are compactly arranged cells and these are homogeneous, means all cells are uniform, all similar type of cells are present. All the cells are of same type. Then as the anther develops, each cell will undergo meiotic division. From this microspore mother cell, each cell will undergo meiotic division. Meiosis means reduction division, and they forms microspore tetrads. Tetrads means 4 cells will be there. So each of this cell from this porogenous tissue, each cell will undergo meiotic division. So each cell of that is called as pollen mother cell. That is the mother cell of pollen, because from that cell is getting divided and it is forming tetrads. So that is the mother cell. So it is called as pollen mother cell, PMC, pollen mother cell, or microspore mother cell. So that is the mother of microspore, right? So microspore mother cell, or it is called as pollen mother cell. And the formation of microspore from a pollen mother cell, it is called as microsporogenesis. The formation of pollen grain from a pollen mother cell, it is called as microsporogenesis. Genesis means what? The formation. So the formation of microspore, that is called as microsporogenesis. Then later what will happen? As a part of development, this anther matures. See, anther will mature. Slowly it will get matured, right? So this anther will mature and it will get dehydrated. There will be dehydration in that anther. Or it may become dry. So that time, this microspore, up to that, it is in tetrad form. Each mother cell will undergo meiotic division and it will form 4 cells, right? So after that, see in this one you can see each cell from the center, microspore mother cell, it will undergo division and it will form microspore tetrad. Here you can see only 3 cells. On the other side, there will be another cell. Okay. So altogether 4 cells will be there. And after dehydration, that each cell will get separated, right? And this tetrads, it will dissociate or it will get separated from each other and it will develop pollen grains. Here you can see this tetrads, it will get separate and it will form a pollen grain. From each cell, a pollen grain will form. Okay, not from the mother cell. After division, tetrads means 4 cells will be there. Each cell will develop as a pollen grain. So what you should do, you should try to connect from the flower. Okay. First, you should remember about the flower. In flower, what and all structures you can see. See, don't try to by heart all these things. Then it will be difficult. You should connect each one with different events. Okay. So first you should remember the structure of a flower, then role of each part, then male part, female part, all about all these things. In male part, male organ, or in androecium, what and all coming. Then you will learn about the filament and anther. Then you have to learn about the structure of anther. Inside anther, pollen grains will be there. So up to pollen grain we have learned, right? Then each microsporangium, thousands of microspore or pollen grains. See, each cell is undergoing division. So each cell will form 4 pollen grains. So like that, so many cells will be there inside this, right? Pollen mother cells will be there. Several pollen mother cells will be there. Each will undergo division and each forms 4 tetrads, right? After division, it will form tetrad structure. So from each one, pollen grains also will develop. So there will be several pollen grains. And when it is getting released, it will get released during anther-dehiscence. See, that is this structure. Dehiscence of an anther. An anther, it's bursting, or that is going to release the pollen grains. So up to this, we have learned the releasing of pollen grains. Now, pollen grains are formed, right? So we have to start today's class with the structure of pollen grain. Now we are going to learn about pollen grain. So up to this, hope it's clear for everyone. So next we are moving to the structure of pollen grain. Okay.
Okay, ma'am.
So in the structure you can see here, in this picture you can see the structure of a pollen grain. Okay. Now I'm going to explain. Listen carefully. Pollen grain are the male gametophyte. Okay. This is the male part. Actually, this is fusing with the female part. Okay. So have you seen pollen grain? If you're touching a flower, like the common flower, I'll explain about the seed. Here you can see the hibiscus flower. See this part, the yellow color. If you're touching that yellow part, which is the anther, this is the anther. Okay. So if you're touching that, some yellow color powder like sprinkles, it will stick. It will stick on your fingers, right? If you're touching that anther, that yellow powder form, something will be there. Some powder forms will be there. That will stick on your finger. They are the pollen grains. Okay. Those are the pollen grains. So in 12th standard practical session, I think you have pollen germination experiment, right? So first, we should know the theory part. Then only it will be easy for you. The structure of pollen grain you should learn. So generally, pollen grains are spherical in shape. They are spherical generally. Okay, they are very small, and with the help of microscope you can see the structure. Okay, while doing the practical session it will be easy for you. And the diameter of a pollen grain, it is around 25 to 50 micrometers. It's not millimeter, it's micrometer, smaller than that. Okay, so it's a minute structure. It's very, very small. So what is the diameter? Its diameter comes around 25 to 50 micrometers. So the diameter of a pollen grain, it's around 25 to 50 micrometer in diameter. Okay, it's smaller
Ma'am,
than
not called millimeter.
It's smaller than millimeter. Here I mentioned it wrongly, sorry. It's micrometer. Okay, very, very,
Ma'am.
very minute. Okay, if you are
Okay,
touching
ma'am.
that yellow powder thing, there will be several thousands of pollen grains on your hand, right? Why it
Okay,
is
ma'am.
producing these many pollen grains, you know? Do you have any idea? Because pollen grains, pollination, it's through wind or something. So many pollinating agents are there, right? So to make sure that a male gamete is reaching the female part, because there may be biotic or abiotic agents, right? So we have to make sure a male gamete is reaching there. So it is producing in more quantity. Okay. So there will be several pollen grains. And this pollen grain, the cell wall of pollen grain, it is called as sporoderm. Commonly, it is called as sporoderm, the cell wall, the wall of a pollen grain. It's called sporoderm. Then pollen grain is having two prominent layers. Okay. This pollen grain, it's having two layers. The outer layer is exine. Here you can see the outer layer. This layer is called as exine. And the inner one is intine. Outer layer is exine and inner layer is intine. Okay. Generally spherical structure and 25 to 50 micrometer in diameter, and the cytoplasm is surrounded by plasma membrane, and pollen grain, it is having two layered wall. Outer layer is exine, inner layer is intine. Okay, inner intine, outer exine. Okay, then next, the outer wall or the outer layer of a pollen grain, it is exine, right? Here the outer layer you can see, this is exine, and exine is made up of sporopollenin. Which material? Sporopollenin, important MCQ, note down. This is, exine is formed of sporopollenin. If you are writing the structure of a pollen grain, you have to write from the outer part, you need to start. Outer is exine, inner layer is intine. Then you are going to write about the exine. Okay, exine, it is made up of sporopollenin and it's an organic material. The sporopollenin is an organic material which is highly resistant. Okay, sporopollenin is organic material. It is highly resistant, and this sporopollenin, it can withstand high temperature, strong acids, alkalis, etc. And no enzyme can degrade this sporopollenin. That is the important point. No enzyme. It can withstand all high temperature, then acid, alkali, everything, and no enzyme can destroy this sporopollen. Okay, no enzyme can degrade. So that is the organic material which can withstand high temperature, strong acid, strong alkalis, and all kind of enzymes. Okay, then because of this property, that pollen grain can survive, right? Otherwise, soon after releasing, it will get degraded or it will get damaged, right? So it can withstand temperature, acid, alkali, then all enzyme. Because of this strong sporopollenin, it can withstand all these conditions. So that a pollen grain can survive easily. So this sporopollen is the most resistant organic material. Okay. So in pollen grains, this exine is having from exine, it is having apertures called germ pores. Okay, germ pores. Here you can see germ pore. See a curve-like structure. In this, the outer coating in exine, it is having some regions. This is called as germ pores. Pore-like structure, germ pores. So in these germ pores, sporopollenin is absent. Okay, in this place it won't be present. Sporopollenin is absent in the germ pores of a pollen grain. In that part, sporopollenin is not present. Other parts and all covered with, or it is made up of sporopollenin. Okay, and this sporopollenin, it helps in fossilization also. How it is helping? Because of the presence of sporopollenin, these pollen grains are well, or these are well preserved as fossils. Because no alkali, just now I told, no alkali, no enzyme, nothing can destroy or degrade this sporopollen. Okay. So it's so hard, so that the pollen grains are well protected from the hazardous environment. Okay. So sporopollenin can protect the pollen grain from all this hazardous environment. And this exine of pollen grain that will exhibit so many patterns. See, this is egg sign. Egg sign is the outer covering, right? That exhibit patterns and designs. See here, you can see pollen grains of different shapes. This is the outer part which is forming different shapes. Here it's spherical. Here you can see some extra growth. These are now egg sign. Egg sign in different shapes. Here also you can see. It's not exact smooth one or round one. Here you can see some kind of design. That is the shape of egg sign. In egg sign, different shapes are forming. And this lot of studies are going on. Different patterns of this egg sign. In this, studies are going on in different types of plants or flowering plants. Different egg sign patterns are seen. So, about this, studies are going on. So egg sign is the hard outer layer made up of sporopollen. This sporopollen is highly resistant organic material. It can withstand high temperature, strong alkali, strong acids. Then no enzyme can degrade sporopollen. As far as no enzyme can degrade this sporopollen. An egg sign, it is having apertures called germ pores. Here you can see the germ pores. And in this germ pores, sporopollen is absent. Clear? And pollen grains, they are preserved as fossils. How? Because of the presence of sporopollen, it can withstand all hazardous environmental conditions. So, it can be preserved as fossil also. And this egg sign, that shows different patterns. Okay. So, studies are going on in this work. So, different patterns and designs of egg signs you can see in different types of pollen grains. Okay. Next is now egg sign over. The outer layer egg sign is over. Next we are moving to intine. The next one. Next layer is intine, the inner wall. Okay. Intine is the next layer of pollen grain. That is the inner wall of pollen grain. It is called as intine. Inner wall, intine. And intine is very thin and it is a continuous layer and it is made up of cellulose and pectin. Intine is made up of cellulose and pectin. Intine is the inner wall. It is thin and continuous layer you can see here. But egg sign, here germ pores are present, right? It's not continuous. In between germ pores are coming. But intine, the inner wall which is thin, it is continuous layer. There is no disturbance. Okay. It is a continuous layer and it is made up of cellulose and pectin. Clear? Next is
Yes, ma'am.
inside this intine. Here you can see intine, right? So inside this intine, you can see cytoplasm. Okay. So inside intine, cytoplasm is present. Here you can see the cytoplasm. And the cytoplasm of the pollen grain, it is surrounded by a plasma membrane. Here you can see the cytoplasm which is surrounded by a plasma membrane. Okay. Here soon after the brown layer, one black layer you can see. That is intine. Okay. Then after that, this next black color line, that is the plasma membrane, and inside the plasma membrane, cytoplasm. This is the cytoplasm, dense cytoplasm is present. And after maturation, a matured pollen grain. Here you can see this is after maturation, a matured pollen grain will be having two cells. Which are the two cells? One is the vegetative cell. Here in this picture you can make out clearly. The first one is the vegetative cell and the second one is the generative cell. Vegetative cell and generative cell. Inside the cytoplasm, I'm saying inside the cytoplasm it is divided as a vegetative cell and a generative cell. Okay. So in the picture here you can see the vegetative and generative cell. In this you can make out it easily. And the vegetative cell. Here you can see both cells. The vegetative cell, it is bigger, right? Here vegetative cell and generative cell, both you can see, but the vegetative cell is bigger. And see the nucleus of the vegetative cell. It is not round shaped, it is somewhat irregularly shaped. Here vegetative cell, it is bigger than the generative cell and the nucleus is irregularly shaped. It is irregular. And this vegetative cell is having so much of reserved food. Okay. Reserved food material is present in the vegetative cell. It's not for us. Okay. Not for human being or not for external sourcing. It's for the growth of the pollen grain. Okay. The vegetative cell and generative cell. Vegetative cell is having lot of reserved food material for the developing pollen grain. Okay. And next is generative cell. This generative cell compared to vegetative cell, generative cell is very small, right? And this generative cell, it is floating in the cytoplasm of vegetative cell. This whole, the cytoplasm, this is vegetative cell, and inside vegetative cell, you can see the generative cell which is floating inside the cytoplasm of vegetative cell. This small one, this is the generative cell, this separation, but it is inside the vegetative cell. So vegetative cell that covers the whole portion. Here the vegetative cell covers the whole portion. Inside the cytoplasm of vegetative cell, the generative cell is floating. Okay, don't get confused. And this generative cell, it is almost spindle shaped. Okay, it is having a nucleus. Here you can see in generative cell also you can see the nucleus and a dense cytoplasm. So what and all we have learned? In a matured pollen grain, you can see two cells. See the structure of a mature pollen grain. It is having two cells. First one is vegetative cell and second one is generative cell. The vegetative cell is bigger one and it is having dense cytoplasm, and this vegetative cell, it is having so much of reserved food material for the developing pollen grain. It's for the developing pollen grain, that reserved food material. It is providing nutrition for the developing pollen grain, vegetative cell. Okay, next is generative cell. Generative cell, it is small and it floats in the dense cytoplasm of the vegetative cell. Here generative cell is also having a nucleus and the cytoplasm is also dense. Like vegetative cell, this cytoplasm is also dense. Okay, and it is almost spindle shaped. Clear? Next is, in almost around 60% of the angiosperms, that pollen grain will be having two cells or two cell stage. While releasing or while shedding the pollen grain, that cell will be in two stage. Two stage cell will be there inside the pollen grain. Normally, in normal cases, around 60% of angiosperms are showing two celled pollen grains. One is vegetative cell and one is generative cell. Okay. That means while shedding that pollen grain, it will be having vegetative cell as well as generative cell in that. Around 60% of angiosperms, they are following this way. And in the remaining angiosperms, around 40% of the flowering plants. So the generative cell divides mitotically. There is a mitotic division in the generative cell in the remaining plant species and all. Okay. Generative cell, not in the vegetative cell, in the generative cell. The generative cell divides mitotically. The generative cell divides or it will undergo mitotic division. That means one cell will divide. Here, this generative, the small cell, the generative cell will divide and it will form two cells. Right? Mitotic division means it is the equational division. Right? So, one cell will divide and it will form another two cells. So, in that 40% of angiosperms, The generative cell divides mitotically to form two male gametes. Generative cells. From that name itself, it is generating male gametes, or it is forming the male gamete. What is the role of vegetative cell? It is providing nutrition to the developing pollen grains. Pollen grains are the pollen grains. What are they? They are the male part, right? So these two, this generative cell will divide, or it will undergo mitotic division, and it will form two male gametes in the remaining 40%. So the pollen grains will be having one big vegetative cell and two small male gametes. So in total, they'll be having three cells, right? In 60% of the angiosperms, they'll be having two cell stage in their pollen grain. Another 40% or the remaining plant species will be having three cell stage in their pollen grain. So while shedding, these anthers or the pollen grains will be having three cell stage in their pollen grain, right? So over 60% of angiosperms shed their pollen grains at two cell stage. In others, generative cell mitotically divide and it will form two male gametes. And the pollen grains of these plants, these angiosperms, they are shed at three cell stage. Clear? The pollen grains in some species it will show two cell stage, and some other species it is showing three cell stage. Okay, and inside pollen grain which division is happening? Mitotic division. But for the formation of pollen grain, which division is there? Meiotic division. Pollen mother cell or microspore mother cell will undergo meiotic division and it will form tetrads. Okay, but in the pollen grain, when it is showing the three cell stage, there meiosis. What is happening? Meiosis. There it is in microspore mother cell, it is the reduction division, meiotic division. It is forming four cells. Here in inside pollen grain, it is mitotic division. Okay, it is forming two cells. One cell is dividing and it is forming another two cells. Means equational division. There is no change in the number of chromosomes. Okay. Next is this pollen grains. Pollen grains of many species of plant that cause allergy. Okay. In some people, it may cause allergy. Parthenium. Do you know about this Parthenium plant? Here you can see the Parthenium plant. Have you seen this? It's a very common plant. Actually,
No,
it is
ma'am.
you might have seen roadside and all, you can see it is a weed actually. So white star like structures means the flower will be like small white color flowers. Parthenium, it's a common plant. It's a weed. It is belonging to Asteraceae family. Okay. So actually it is a weed. It is mainly seen in agriculture fields and all, you can say field, garden, everywhere. It's a common one. And that is a weed that reduce crop productivity because it can absorb nutrients very fast. Compared to that crop, this will be very fast in absorbing nutrients. So that reduces the crop productivity. Okay. And what else I can say about this Parthenium? And this Parthenium is reducing the germination. That can reduce the germination. If you are sowing some crops, some legume plants and all, if you are sowing, that this Parthenium is present there means it will reduce the germination percentage also. Okay, it can reduce, it is having the power of reducing the germination of the crop, and it will reduce the biomass, then growth. It will affect everything because that is having the capacity to absorb nutrients very fast, and this will grow very well in the field compared to the crop. Okay, so it will inhibit the growth of the root. Parthenium is having another way of suspending the growth. Okay, it will inhibit the growth of the root. Like that also it will destroy the crop. And this pollen grains, just coming back to our point, the pollen grain of this Parthenium that may cause allergy. Okay, some skin allergy it may cause. So pollen grains that may cause allergy also. And these pollen grains, they are rich in nutrients. Right? Pollen grains, it is having vegetative cell, generative cell, I said. So for the growing pollen grain, it will be having a nutrient source inside that pollen grain itself. So the pollen grain, it is rich in nutrients. So the pollen tablets are made from pollen grains because it is rich in nutrients. Here you can see these are pollen tablets. These are made from pollen grains, and these pollen tablets, they are nowadays using as a food supplement. Okay, we are consuming it as a food supplement. Same like pollen tablet, pollen syrups are there. That is also available in the market. So study says that the consumption or the intake of these kind of pollen tablet or pollen syrup,
Ma'am,
this will
pollen is used for
sorry.
medical.
That's what for see it is rich in nutrients I said, right? So these tablets and syrups that the studies are going on. In that studies, it is saying that the performance of the athletes, because it is energy, it's like an energy drink. It's providing more energy. So that can increase the energy source. So for the performance of athletes in that kind of wherever we require more energy for that kind of and this race horses are there for that and for athletes and all we can provide this energy source, the syrup and the tablet for more energy for the to increase the performance. Okay. So once this pollen grain is shed, that should reach where it should reach. Now pollen grain is ready. Where it should reach now? It should reach on stigma for the other steps, right? For fertilization, this is the male part. It should reach the female part for fertilization, right? So the pollen grains, see here in this picture, you can see this is the yellow part of hibiscus. And if you are touching this, I said yellow color dust-like forms will be there. That and all the pollen grains. Here you can see different shapes of pollen grains. The pattern studies are going on the pattern of hexine, I said. Here you can see different shapes of this pollen grains. Okay, in this picture. And the pollen grains, once it is shed or once it is out, those pollen grains should reach exact place for fertilization. That also before losing its viability. Because that pollen grains can't survive for more time, right? It will lose its viability. So to bring out fertilization, that pollen grain should reach on stigma. Where it should reach? It should reach on stigma before losing the viability. So the period for which that pollen grain is or the pollen grain, it retains the ability to germinate when it is landing on stigma. The pollen grain will land on stigma. Then that pollen grain should germinate and it should reach near the ovary, right? For the fertilization. That pollen grain should reach near the female part. So for that, before losing the viability, all this should happen. So that period, in which period the pollen can, up to a certain period, a pollen can survive. That period is called as pollen viability. It is called as pollen viability. So this pollen viability, it is variable. Means up to a limit, it is depending upon humidity, temperature and all. It is highly variable. In the case of wheat, rice and all, pollen will be viable up to 30 minutes only. It will be viable only up to 30 minutes. This pollen grains will be active or the active period of that pollen grain, it is called as viable period or the viability. That rice, wheat and all pollen will be viable up to 30 minutes after releasing. If it is getting released up to 30 minutes, that pollens can survive. But in Leguminosae, Solanaceae, Leguminosae means green gram, cowpea, all these things and all coming under Leguminosae, legume plants. Okay. Then Solanaceae, which are all coming under Solanaceae? Chili, tomato, all these are coming under Solanaceae family. So in that family, then pollen is viable up to several months. That viability, pollen won't get degraded. It will be viable up to several months. Okay. So we can store this pollen. So you know that artificial insemination in animals and humans and all. For artificial insemination, the storage of semen or storage of sperm is done, right? For that, storage of semen or sperm is done. So in the same way, in plants, that is in the case of animals. But in the same way, it is possible in plants also. The pollen grains, which is the male gamete or the male part that we can store. Okay. How we will store this? With the help of liquid nitrogen. With the help of liquid nitrogen, we can store this. This is called as the storing of this under very low temperature. Very low temperature. In liquid nitrogen, the temperature will be around minus 196 degrees centigrade. Okay, minus 196. So it is in a very low temperature. Okay, so it is called as cryopreservation. This method is called as cryopreservation. MCQ, note down, cryopreservation. Storing under very low temperature. It is called as cryopreservation. So we are using liquid nitrogen and all for a medium for cryopreservation, right? So we can store like this several years we can store. Not only this one, we are storing the plant material, everything for DNA isolation and all. See, one crop we have taken up sowing now and we need to store it for another 2 years, the DNA or something, we can store in the same way, cryopreservation. Storing under very low temperature so that there won't be any changes in that. At very low temperature, for an active DNA or active material, there should be a normal temperature. So if you are lowering the temperature, there won't be any active process. So in that temperature, it will be stable or no changes will happen in that. So such low temperature, that method is called as cryopreservation. So such stored pollen grains can be used as pollen banks. Pollen banks means where we are storing, and later for any other studies we can utilize, just like seed bank. Seed bank means all old type of seed, wild varieties, all these things will be storing there. And whenever we need for further studies or crop breeding process, we can take that and use it. So in the same way, this pollen grain also we can store. And what we will do after storing this pollen grain? We can use it later for making hybrids and all, right? That time this type of pollen grains or this type of plant is not available means we can use this pollen grain and bring out the same character in the new generation also, right? To make the new hybrids, we can use this pollen grains, right? So is it clear? The shed pollen grains they have to land on a stigma before losing viability. So the viability period of a pollen grain, it is different or it is variable. It depends upon the temperature and humidity. Okay. The viability of pollen grains of some cereals like wheat, rice and all, that is around 30 minutes. But in Leguminosae family means legume plants and Solanaceae family like chili, tomato and all, the viability is for several months. Clear? Is it clear? So the structure of pollen grain, now it is over. I think it's time up. Now I don't have time to explain the structure of this gynoecium. So androecium, the male part we have completed. Okay. Do you have any doubt in that? I need to repeat
No,
anything?
ma'am.
Is it
No,
clear?
ma'am. Yes,
See,
ma'am.
you have any doubt means you have to ask. Okay. And daily, whatever I am explaining, at least one time you should read. Then it will be very easy for you people. It will be very clear. Okay. So every day read this. In the next class, we will be explaining this one, the gynoecium, the female part. Next class, we will start that. Now I'm starting also. I think I can't complete. I can't even half also. I can't explain. It's almost time up. So read this, practice the questions, read it properly, and after completing each chapter, practice whichever questions I'm giving. Nearly minimum 200 questions I'm giving per chapter, right? So practice all those questions. Then previous year questions also you have to practice so that you will get a complete idea of this one. And if you have any doubt, you can share with them. I'll collect that doubts from them, and in the next class, I'll be explaining that. Okay. Okay. So today's
Okay, ma'am.
class over. Thank you so much.
Thank you, ma'am.